we ran the measurement, and then we analyzed all the data, and we wrote the paper within a week. It doesn't mean that the paper got published in another week. It took a little bit longer, but we really enjoy working together. He's a great scientist. He's a close friend, and I'm very happy to have you today here in the workshop, Suman.
Thanks. Can you hear me?
Yeah, perfect.
Yep.
Okay. Let me. All right. So, and you can see the screen, right? All right. So, when I originally planned on giving this talk, it was supposed to be applications of microscopy. But then, after talking to Lionel, we thought it would be a good talk, a slightly different talk based on metabolic optical imaging. So it's going to go a little bit more into the history of the optical imaging, optical metabolic imaging, or metabolic optical imaging. And we'll see like how phasor fits into that category. So of course, Enrico and Dave is there. Alex already talked about some stuff, so I probably won't have to go into too much detail about the phasor. So I'll start with a little bit of historical perspective. And the front picture is where I am. That's my university now, where I work as an assistant professor. And thanks Lionel for this really nice introduction. Yeah, the paper took less than a week to write, maybe. It took a week to get accepted, but it took 3 months to come out, or 4 months to come out. So that's a different issue. But anyway, going back. So most of the optical imaging, optical metabolic imaging, you can actually get the roots to this paper from Beaton Chance in Science in 1962. And it's looking at intercellular oxidation and reduction states in vivo. And what they're doing is they are looking at mouse, anesthetized mouse in kidney and brain. And this is not a fluorescence imaging experiment. This is 1962. So it's a more of a, it's a spectrophotometer experiment, right? It's with the objective. And what they're looking at is they're looking at what happens to the kidney and the brain under different stimuli. And one of the things they saw, and it's kind of important, is they looked at DPNH, which is basically NADH, it's the old nomenclature. And they looked at between the perfused kidney slice, which is shown in here, and the unperfused kidney slice, there is a change in the spectra. So that there is something happening in the spectral range over there that you can see. And this is all UV excitation. The other important thing that's in the paper is if you change the breathing system, so you're changing from oxygen to nitrogen, and going down here is actually increasing the fluorescence intensity. You see, based on the different gases that you are actually giving the mice, you see this change in fluorescence intensity. So there is also a change in the intensity and spectra based on how you are changing the situation, right? And that sort of started the field, but the imaging started coming along after a 2002 paper from Watoev's lab, where they did two-photon excitation spectra of the NADH, NADPH, FAD, and LIPDA. So these are the excitation spectra, and you can see around 700 to 750, you can excite NADH, and FAD, on the other hand, has a larger excitation spectrum, but you can also excite FAD at the same wavelength range. And based on the microscopes that we have, based on the lasers that we have, titanium sapphire, this is a proper place to excite. And the emission spectra, you can separate out the NADH emission spectra and the FAD spectra by filters. That sort of started the field. And the question then became, okay, what can we see and what can we do? So a few of the figures that I took for this presentation are from this review from Kyle Quinn in the University of Arkansas. And what we can see is basically this, right? We're looking at, it's a very simplified diagram of metabolism. And you are looking at NADH. You are looking at mitochondria or cytoplasm or outside in the nucleus. And two of the components, NADH and FAD, two of the coenzymes, are part of most of these metabolic pathways: TCA cycle, PDH6, glycolysis, lactate dehydrogenase. And if you look carefully, the NADH is brighter and NAD plus is dark. The reverse goes for FAD. FADH2 is darker. FAD is bright, because these are the fluorescent species you can see. FADH2 and NAD plus are dark, so you cannot see. So the idea is, can we use the NADH and FAD to actually see what is happening in metabolism? And that gave rise to this metabolic optical redox ratio, which is basically an intensity ratio of the FAD by FAD plus NADH. Now I have to tell you that there are different definitions of this. There are different people define it differently. Some people define it FAD by NADH only. Some people define it NADH by FAD. So you have to look at it carefully if you are comparing between paper to paper. But the idea was, okay, if we can do this, and can we actually correlate it with the actual concentration of the NAD plus from NAD plus by NADH, which is known to be a metabolic index. And that basically is possible with 2-photon excitation. If you do 1-photon excitation, you are going to put so much power in the system that it's going to fry the sample. So especially biological system. So the 2-photon excitation lets us do that, and we can look at the lifetime and the quantum yield and the intensity to study metabolic changes. And so this is basically the way what we can see and what we can do. Now, of course, this requires validation. And one of the major validation comes from the intercellular cofactor ratio based on liquid chromatography mass spectroscopy, which has been gold standard in the system. Of course, it doesn't give you cell-to-cell variations or intercellular variation, but this has been biologically the gold standard. Now, one of the papers that talks about it, they looked at human bone marrow-derived mesenchymal stem cells, and these stem cells are going into different types of differentiation. So there's adipogenic, osteogenic, and MSC propagation, and trying to see what happens after the induction, right? So if you look at the optical redox ratio as defined by here, FAD by NADH plus FAD, and if you look at the LCMS ratio, NAD plus by NADH plus NAD, and these three colors are three types of differentiation. That's what they are. You can see a very good correlation between the optical redox ratio and LCMS. This correlation was much less for if we measure the LCMS of FAD by NADH plus FAD. Now, in terms of imaging, and this is the paper, you can look at in MSC propagation, osteogenic and adipogenic, you have very different redox ratio. And you can see the subcellular differentiation here, and come back to that in a second. But also, another paper that looked at it is from Kyle's lab again, which looks at optical redox ratio and in precancerous epithelial tissue. And I'll come back to this precancerous epithelial tissues again. So these are epithelial tissue which has different types of, they're differently modulated because some of them has higher chances of getting cancer. And in this case, the LCMS ratio and the optical redox ratio are highly correlated in both definitions. So there seems to be a correlation between the LCMS measurements that has been done historically and the optical redox ratio. Now, of course, you do not get the intra and intracellular variability that you can get with LCMS, in any optical imaging. That's why we do most of the imaging, because of this much more detailed information that we can get. Now, another validation is, of course, which is the gold standard now in a way, which is with OCR, oxygen consumption rate, which is basically the rate in seahorse assays, which is everybody uses. Again, seahorse assay is a solution measurement right on top of the cells, which measures the not single cell level. It measures whatever happens to the whole system. Now, in this case, again, it's Nirmala Rajaram from University of Wisconsin and her lab, and they have been looking at comparing it with what they call in OCR, which is the oxygen consumption rate, and proton production rate, which is Again, a method of looking at metabolism. And what they did is they blocked the cycle of mitochondrial metabolism by different pathways. So they added oligomycin, which inhibits the ATP synthase. Then they added FCCP, which takes out the uncoupling from NADH, so they uncouple the transport chain. And then rotenone and antimycin, which blocks complex 1 and complex 3, and combined together, they basically block the, change the metabolic pathway. And this is basically what they saw in NADH and FAD. These are just intensity images, but these are redox ratios between 0 to 0.6. But the main thing is here, if you look at the redox ratio, oligomycin decreases the redox ratio, FCCP increases the redox ratio, and antimycin A and rotenone again decreases the redox ratio. It's the same exact behavior that you see in OCR. So this is another validation that whatever we have been seeing with this particular definition of optical redox ratio, it actually validates very well within OCR. Similarly, they have looked at normoxia, so normal oxygen concentration, and then they looked at what happens if you put them in hypoxia and then reoxygenate it, or what happens to the optical redox ratio. So again, this is optical redox ratio. This graph is in OCR, 4 different types of cell line, in both in optical redox ratio of 41, 40.07, it goes up. FARN, it doesn't, and NR, it actually goes down a little bit. Same exact behavior here. And what they realized that by looking at these, they can actually look at the metastatic potential of the cells, and these cells are more metastatic, so they have a higher change. So this is another sort of another validation for the approach to BOR. Now, this is all intensity-based measurement, right? So it's a ratio of the intensities, whatever definition you want to use. And then we do lifetime experiments. So the question, of course, becomes, why do we do lifetime experiments? So you have to think in a way that intensity of any of the species depends on sum of a fractional intensity, which is the ratio of the quantum yield multiplied by the fraction of that particular species. Now, if it's just one species, FAD and NADH, it works fine. But the problem is, it's not one species. Again, I'm going to go back to this. And if you look at it, there is protein-bound NADH, free NADH, which has very different quantum yield. Free FAD, protein-bound FAD, I couldn't find the number for this, but they also have very different quantum yield. And if you do think about that, then whatever you are seeing in FAD or NADH is going to be completely dependent on what is the fraction of each of these species at present, which makes this correlation sort of difficult to do. And again, in free NADH, the 2 rings up here collapses, which are close proximity, so that decreases the quantum yield, and that's shown is 0.02, at a very low level. And when they are actually further apart after binding to protein, they go away and it increases the quantum yield. So that becomes very important. So we decided, okay, we will do we didn't decide, it's been decided before, lifetime experiments. And lifetime, after all the talks that Alex did and Indico did earlier, I don't have to say anything. It's just basically the time the molecule spends on an average on the excitation state, and you can get a decay in the old traditional way, and you can find the lifetime. And I'm going to go a little bit, before I go into Phasor, I'm going to talk a little bit about just the TCSPC, the time correlated single photon approach, which has been used in the biological system for a while also. But the big thing is this is independent of concentration, so we can actually calculate the lifetimes of these species, and maybe we can correlate that to the optical redox ratio or the metabolism. So the first paper that sort of started that is from Damien Bard and Ramanujam. I don't know how I'm saying that, but they are looking at MCF10A breast cancer cells, and they are looking at how many cells are there. So they're looking at a level of cell density in a sense. And you can see that the lifetime in low cell density to high cell density changes. And what they saw, they fitted this exponential decays by 2 exponentials, and they calculated the free and bound lifetime. But what's more important here is the free to bound protein ratio. So that's the ratio when the number cells are 25,000, this is 100,000, and this is a million. And you see this really large increase in the free to bound protein ratio over the number of cells. Now, if you go to same system, but you start the NADH, so you're decreasing the metabolic factor, you actually don't see as much of a change. And if you do it with potassium cyanide, which is which would move it to anaerobic metabolism, you see this system basically behaving similar to serum stamp. So this gave us the way that, okay, maybe looking at lifetime would help us to understand metabolic changes. And again, this is another paper that deals with the precancerous epithelia, which I talked about before. So these are different cancer cells. And here, this row is normal, then these are low grade, and this is high grade, and similar is true. This is the optical redox ratio. The definition is different again here. And you see the redox ratio changing, going down, but also changing depending on how much precancerous cells are. Same thing is here. You see average NADH lifetimes, and this is only average lifetime. It's changing this way or this way and this way, right? So by looking at that, they found that the contribution of protein-bound NADH between the normal, low-grade, and high-grade cancers or precancerous epithelia changes. So this is decreasing. And so the main point of the paper is that the increase in free cytosolic NADH that occurs in anaerobic glycolysis not only decreases the optical redox ratio, but also produces a decrease in the mean NADPH. And this is from Melissa Scala when she was a graduate student, I think. And it was published in PNAS in 2007. Now, another paper which looked at it in more detail is, and in a very different organ, is brain. So most of the publication in this field comes from cancer, but this is looking at brain, and basically they are putting topical reagent, and they are looking at what happens to the metabolism. So they have their control, and then they are blocking the glycolysis by 2 different drugs, D-glucose and iodoacetic acid. Then they're blocking the Krebs cycle by, again, 2 different ways. Electron transport chain by rotenone and potassium cyanide, oxidative phosphorylation by FCCP, and they have a focal seizure model because it's looking at actually that. And that's just a map of where different things are getting blocked. But if you look at the average lifetime, and this is again just looking at average lifetime, between the samples where there is glycolysis blocked, it has a longer average lifetime. But if you block the TCA, the lifetime goes away. If you block the electron transport chain, it goes to the lowest. And then if you uncouple them, by using HCCP, it actually goes back to the level here. So that's sort of, again, another validation that this works. A validation compared to OCR from Seahorse assay was shown here. So there's a lot more data here. I'm not going to talk about MCR because it's different. But what you should notice is the free to bound ratio, redox ratio, and OCR. And free to bound ratio here in NADH is measured by lifetime measurement. So if you free to bound NADH ratio goes up here, the way it is defined, redox ratio should go down, OCR to go down. And if you look at the mitochondrial stress test by oligomycin, FCCP, and rotenone, you see oligomycin increases the free to bound NADH ratio, these 2 goes down. FCCP, it doesn't change a lot, decreases slightly, these 2 goes up. Rotenone, this goes up, this goes down. What about glycolysis stress tests? Glucose, oligomycin, 2DG, you look at the glycolysis, and again, if this goes up, this goes down, this goes up, this goes down. So there's a very good correlation between different types of measurement, and you can say that, okay, we can see it. Similar things, and because I haven't introduced Phasor, and I was not sure how the talks are going before me. So if you look at Kiara's Stingray and Karapet, their paper on wind signaling, you can see that exactly similar thing was done with comparing the phasor to all these drugs, and it's in EMO journal, published quite a few years ago now. Now, the lifetime imaging and phasor approach. So you already heard this. So let's say if we have a 256 by 256 image, we have 65,000 points. So fitting each of the points individually is very cumbersome. And that's one of the reasons we do phasor. So this is just looking at a laser pulse and then the decay. And what we do is we transform them by cos and sine. The Fourier transform gives us the phasor plot. And by looking at, if you have a very fast decay, you can see the sine multiplication would be basically 0 and cos multiplication would be higher, close to 1. So it gives you the value up here. But if you have a very slow decay, which basically didn't decay here, you would get, or infinity lifetime in a sense, you will come here and something along the middle would come somewhere in the middle. Now, one of the biggest thing about the phasor is the reciprocity principle. So what I mean by that is you have your fluorescence intensity image and you transform them to the phasor. Now, once you have transformed them, every point of this image gets transformed to a point in the phasor plot. Now, you can use part of this phasor plot to select what kind of lifetime signature or phasor signature they have. And you can see where in the image that goes through. This is very important because let's say if you have a very few pixels of a very different lifetime, and that would be a very separate position here, and then it will allow you to look at that, which conventional fitting approaches would not allow you. The other thing is you can also look at the intensity image, select out a portion, see what their phasor signatures are. And because the reversibility of this whole process is very fast, and you can basically look at biological system and complex biological system much better than a fitting approach would allow you to. The other thing that actually is very important, and Alex has gone through this quite more detail, is the algebra of phasor. So if you have two components, and if you have a point in the image which has both of these components present, the fraction of each of these components will determine where it will be, and it will always be in the line that is joining the two. Now, based on that, you can actually calculate the fraction of, so if you have something which is distributed like this, we can actually change it to a fractional intensity plot, and we will look at that. So now the procedures, you do the image acquisition, you calibrate the acquisition. The calibration can be done using a fluorescence dye. So let's say this is the position of the fluorescence dye. What we do is, and when the system is not calibrated due to delays and other electronic stuff, it will come, let's say it comes here. This is the original position. So you first correct for the angle, and then you correct for the modulation. So you push it back here. So you know how much of this change you need to do for correcting other sample. You can also use a scattering sample, which has a lifetime of 0. So it will go the reverse way. And once you have calibrated, you can reference all the samples. And then you can do masking, image filtering, and intensity selection by looking at different areas, different intensity areas, and different types of system we have seen. We have looked at biological fluorescence, which I'm going to go in a bit more detail soon, freight, concentration, pH, and you can do different types of analysis. So I don't know if Alex talked about it, but we have gone up to 4 components now. Now, reasons of doing FLIM is fit and compared to other phasor, compared to other, is fit free, is visual in nature, so it's very intuitive. The decays and phasor properties assigned to the molecular species and not lifetime, so that's important. It's an image, so most of the analysis procedure you can use for image analysis actually works on the phasor also. And the other thing is filtering without changing the resolution of the image. So what I mean by that, let's say this is your phasor diagram. You can do a filtering by assigning a median filter and assigning the average phasor to the center of that filter. You can also do a smoothing. So if you do a median filter, you decrease the spread but it doesn't change the original image. The original image still has the same exact resolution. In comparison, if you do a smoothing filter, original image loses its ability to look at that, right? And then the reciprocity principle, which we talked about a little bit ago, and the linear addition properties of the Phasor space. So based on that, how do we do metabolic imaging? We use protein-bound NADH and free NADH. And free NADH has a shorter lifetime, which appears here. Bound NADH has a longer lifetime, appears here. If it's just these two species, the distribution would always appear along this line, which is called metabolic trajectory. And the shift along this trajectory tells you how much, what kind of metabolic changes is happening. The first paper that was looked at from Enrico's lab was from Kiera, which is looking at neural stem cell differentiation. And if you add reducing conditions, so by potassium, adding potassium cyanide, it goes more towards free NADH. If you add oxidizing conditions, so hydrogen peroxide, it goes more towards bound NADH. By understanding that, they also looked at glycolysis. So if you add high glucose, it goes towards free NADH. And if you use sodium dichloroacetate, which is push it towards oxidative phosphorylation, you see it's more going towards bound NADH. So now we have a way to look at changes between the glycolysis and oxidative phosphorylation, at least a relative fraction of that. So Rupsa, who was a graduate student in Enrico, and this is from Enrico's lab, they looked at in a collaboration in Huntington mice, and they're looking at metabolic reprogramming in astrocytes, and they're looking at striatum and cerebellum. And by doing that, what they did is they looked at what happens with this Huntington disease and how the metabolic imaging NADH changes with Huntington disease. So they looked at Huntington and wild type disease, young mice comparatively 20 weeks and old mice greater than 80 weeks. And if you look at the striatum, you see a huge difference between the wild type and the young Huntington mice. But old mice, there is not much change. This is sort of shown here. In striatum between the wild type and the young, in the Huntington disease, there's a huge change in the bound NADH fraction. However, this is much less in old mice. But in cerebellum, which is resistant to Huntington disease, you don't see these changes. Now, in my own work, I wanted to give a glimpse of it. So this is work with Paulo Sassone-Corsi, who has sadly been passed away, from Center of Epigenetics and Metabolism, and Lorena Aguilar, who is in UNAM in Mexico, and of course Enrico. And this is looking at spatial dynamics of SAT1. Now what we are looking at is we are looking at gene expression. So we're looking at gene plasticity and how this works in presence of different writers, readers, and erasers. So the writers modify the either the histone or the DNA. The readers read it and figure out what changes in the gene expression or the plasticity needs to be done. And then the erasers actually takes whatever the writers wrote away, so new things can be, or modification can be made. So SAT1, which is a protein, which is also a deacetylase. It's a clock protein which looks at our sleeping cycles and stuff like that. But it's also a deacetylase, which is an eraser. And we figured that it has to bind to this for its eraser activity, which means its diffusion would change based on the activity. So we can look at that SAT1 activity by diffusion. And to do that, we used a technique that Enrico, me, and Luca Lanzano have done before. So this is just simulation of beads moving very fast, medium, and slow. Now, it's 256 by 256 point. If you want to get a individual decay curve, you have to, or fluorescence correlation spectroscopy at each point is going to take 10 seconds at each point. So that's not even possible. So what we did, we looked at very short intensity decays, and we looked at, and we converted them, after we calculate the autocorrelation function of the short intensity decays, we converted them to phasor plot actually, and we saw that. So this is sort of what you would expect in single exponential, but this is what you see when you do a very short lifetime. So this is 64 multiplied by 20 microseconds. That's short of a lifetime. And then you can transform them, and you can use the phasor to select out slow and fast diffusion based on the color. So slow would be up here, fast would be up here. Now, we can also look at what happens to the NAD plus metabolism in compartmentalized nucleus. So this is nucleus, and a salvage cycle, which is the part of nuclear cycle, involves NAD, NAM, and NMN, and different enzymes. And we wanted to see what happens, and if we can see this, these changes here. So we, of course, look at phasor FLIM of NADH to look at the nucleus. So going into the actual expectation, idea was we would modify the salvage pathway, and SAT1 would become active, and we should see that. But when they are active, we would expect the SAT1 slow SAT1 diffusion, and when they're inactive, we would see a fast SAT1 diffusion. We will also see a higher ratio of free NADH when SAT1 is active, and lower ratio of free NADH when it's non-active. And so to do that, what we did is we took the salvage cycle and blocked the SAT1 activity by mutation or drugs. We also added FK866, which is a drug that blocks this enzyme, which is the rate limiting step of this process. So these are our direct control. But in our metabolic control, we added NAD and NAM and see what happens to this. So to show the data, so this is basically the FLIM data from the nucleus. So you see the images, then they are marked for only the nucleus, and then they are looked at the lifetime. And between wild type and SAT1, you see there is a change, a SAT1 negative cells, so inactive cells. So this blue is the wild type, red is the SAT1 negative. It goes more towards bound NADH. However, if we add EX527, which would block the SAT1 activity, we see slowly it's going away the similar manner as the SAT1 negative cell. However, if we add it to SAT1 negative cells, which shouldn't matter, it doesn't. We don't see any changes. And SAT6 negative cells, which is not in nucleus, which is not a part of this, there is no change compared to the wild type. Now, what happens to our diffusion? So slower diffusion is going to be shown in the purple, and higher diffusion coefficients are going to be shown in the more red and orange. And you see the SAT1, these cells, which is SAT1 is tagged with EGFP, but they're positive. You see this purple flecks all over. However, if you make the SAT1 negative, make it inactive version of it, it goes away. So, this fraction of this slow, so the bound fraction, which is actually active, is going away. Similarly, if we do the same thing with the drug, as a function of the drug, how much time the drug has to interact, it starts going away again. So this was our direct control experiments, which correlated our FLIM data with our diffusion data. Now we went back and looked at the indirect inactivation. And we did it by blocking again NMPT. We added NAM, which is actually inhibitor of SAT1, and by adding NAD+. So in non-treated NAD+ and NAD over time, you see there is a change, but then the change slowly goes away, which is shown here. However, NAM, you see the reverse change, which you expect from this, right? And in SAT1 negative cells, there is no change. However, if we do FK866, you see basically similar value as adding NAM, it's going away. But SAT1 negative cells has non-significant changes. We looked at similar things with diffusion. So again, we add FK866 over time, it stops the SAT1 being active. So by blocking the salvage pathway, we are making SAT1 inactive. On the other hand, we took these inactive cells and we treat them with NAD+ or beta NMN, and you see the activity starts coming back. So this was sort of a fast direct experiment where we looked at how metabolic changes affects the activity of a deacetylase, and on the reverse, activity of the deacetylase changes the activity of, or shows a change in the metabolism. So that got published a few years ago, but there is a word of caution, and this is sort of the papers that Lionel was talking about before we started. So if you look at the publications on Phasor, you see different trajectories for NADH, different positions for bound NADH. Some people put it here, some people put it in the middle. Why? Well, there's one of that comes from the difference in the bound NADH lifetime, because depending on what protein it is binding to, it can have different lifetime, and that would change the position here. But a bigger problem seems to be how this data is analyzed. And part of that is how this data is transformed. So I'll come back to this in the next slide again. So basically, most of the people takes data at, on a Baker and Hickel card based on TCS-PC approaches with a gain of 5, which is about less than 10 nanoseconds. So it's about 8 nanoseconds. Now we are trying to take this and we are trying to fit the decay that appears here, which didn't decay to 0. In that case, the fitting doesn't work very well. Alternatively, if you're trying to transform these to a phasor, this is a non-repeating signal. So Fourier transform is a repeating signal. So that doesn't work very well. So what Enrico proposes, and we showed in the paper, is if you're trying to do a phasor transform, you should take a much lower gain, so gain of 2, which is 25 nanoseconds. You select a 12.5 nanosecond window out of that, which is actually repeating pattern, and you could convert that. That's the proper transformation. And so these sort of work got Lionel and I published in 2 continuous papers. One is to look at bound and free NADH. The other one is the proper transformation. And because these differences, you would have different metabolic trajectories from different papers. So it's worth looking at it with a sort of a cautious mind. Now, this effect of proper transformation can be seen very easily. So this is FLIMBox, which is frequency domain, which we use often. If you do Baker and Hickel gain of 2, you basically get the exact same results that you get in a FLIMBox. However, if you do the gain of 5, you see a very different trajectory, and it even goes outside universal cycle. And this is sort of what we saw this is just a combination of these 3 together. In terms of actual cells, if you do FLIMBox or Baker-Hickel gain of 2, we basically get similar distribution. However, if you do gain of 5, we get a very different distribution. So if you're comparing this with this, you're not going to get, the relative fraction might work, but the actual quantitative information will not be around. So what about fixation? So it has been shown that MDA, so this is a triple negative breast cancer cell, MCF10A is a non-cancerous epithelial cell. And if you look at the live and fixed sample, you basically see the same behavior, different extent, however, similar behavior in lifetime of NADH. This doesn't work for FAD. FAD completely changes, but in NADH it works. So this is again control sodium cyanide to deoxyglucose coming down, going up. Control sodium cyanide to deoxyglucose in live cell coming down and going up. How about each pixel to pixel? So you can take the same area of the live and fixed, and if you look at a co-localization histogram, you see a very high degree of co-localization. It seems to say the exact same thing that is present in the live cell shows up in the fixed sample also. In Phasor, yes, it get transformed. Of course, the lifetimes are different, so there's a shift. However, it's still along the metabolic trajectory, so we can compare between similar preparations. And finally, if we look at mean lifetime distribution on normal pancreas and cancerous pancreas, it will shift to the lower lifetime, and that's exactly what they have seen. Now, in a few minutes, I'm not sure how much I have, but I'm gonna just introduce this. What else can we see? We can see oxidative stress. So this is Rupsa's work, and it's basically transmission and these are transmission fluorescence images. She was dealing with cells which is fed with oleic acid, and she saw this different behavior which is different than the metabolic trajectory. So you can just color code them. However, when she did coherent anti-Raman spectra or third harmonic generation, which looks at lipid droplets, you can see this signals are co-localized within the lipid droplets, this signal. And this is coincident, as shown by Raman, with the triglyceride peak. Now, in terms of oxidative stress in human pluripotent stem cells, so this is normoxia and this is hypoxia going along the way. And as you can see, this is there's a change here. So this is just the NADH. Don't have time to go into it. But in terms of oxidative stress, you start seeing that, okay, with more and more hypoxia, the more and more this LLS species is presenting. You can see it in the phasor plot, you can see it's a quantification. But of course, this is sort of a broad quantification. So in vivo, and I wanted to work a bit more. So we first created this, that was a few years ago, maybe last year, fraction intensity contribution in a 3-component system. So the idea is, if we have only 2 components, this would be along this line. However, if we have a third component, based on how much this third component is, it's going to drag it towards that component. Now, it would always be within these 3 points. We looked at that. What we did is but if you can think about, you can select a point inside this, you can project a line from here to the original metabolic trajectory, and you can do that for the whole distribution, and you can transform it to a distribution. So by doing that, we can calculate what's the fraction of this component or what the fraction of these components. So by that. And then based on that, we looked at this high fat, high cholesterol Western diet and normal diet back in some years ago. And this is normal diet, but with the fat diet, you see the changes in the liver. These are all fat droplets. But we can do the quantification and we can see how much is this long lifetime species is present. This is work with Alex. I'm not sure has time to go through, but this is basically, we converted that 3-component to now 4 components. So these are 4 different dyes mixed in, and you can see they're always along different lines, and we can actually calculate their fractions. We have looked at cells, which has 3 different nuclear dyes again, and we can, individual cells, we can calculate their fractions. They're only going to be 1. However, when they're mixed in different amounts, we can get those fractions. This seems to be really interesting to use in large biological system where every small area of the image gives you a different value. So we have a much higher statistics to get into. This is blind resolution. Alex already talked about that, so I'm gonna skip that in the interest of time. But again, we basically were able to get the numbers that we have done previously with knowing what the components are, but in this case, without knowing any of the components. Finally, to acknowledge LFD, where I did almost all this work. Some of the work continued after I moved to Georgetown as my independent position. Enrico, of course, nothing would have been done without him. All of this was his idea, of course. And whenever we come to him, he says, we can do it, don't worry. Lionel, I don't know how many projects we have worked on so far, and hopefully that continues. And this is just because he loves Ramones. And then Alex, Belen, and who are partners in crime in the last couple of papers, and then Sasha, we have been working together since I joined with the diver, and I'll talk about it in the next time in the diver talk.